312 patients. In a patient population that represents 1 to 2% of non-small cell lung cancer diagnoses, across 45 sites, in 3 years, Meyer Osei, Arventa Global, Clinical Trial Manager. Patient recruitment in a molecularly selected oncology trial is a numbers problem. RET fusion-positive NSCLC is rare, maybe 2,000 new cases per year in the United States. Of those, many will be treated in the first-line setting with a RET inhibitor that doesn't exist yet. Of those who progress and are second-line eligible, some will be too sick, some will be on a competing trial, some won't consent. Our screen failure rate is 31%. That means for every 10 patients who enter screening, 3 don't enroll. The biggest drivers: NGS result unavailable or inconclusive, the test wasn't ordered, or the sample was insufficient. ECOG performance status too high, the patient has deteriorated between referral and consent. And prior RET inhibitor use, a criterion that became more common as first-line RET inhibitors gained approval mid-trial. We added 8 sites in Japan when U.S. enrollment slowed in year 2. Japanese sites have high RET fusion testing rates and efficient referral networks. They now account for 22% of enrolled patients. Enrollment isn't a passive process. It's active intelligence. Which sites are identifying patients, what's causing screen failures, and where do we need to intervene? Agentforce enrollment analytics surfaces real-time screen failure reasons by site and eligibility criterion. Molecularly selected patient populations, the recruitment mathematics. Clinical trials targeting molecular subpopulations face a fundamental recruitment challenge. The eligible patient population is small, geographically dispersed, and requires molecular testing to identify. For clarity 301, the eligible population, RET fusion-positive NSCLC patients who have received prior platinum-based chemotherapy and have not received a prior RET inhibitor, represents an estimated fraction of total NSCLC diagnoses at any given site. The recruitment mathematics must account for the incidence of RET fusions in NSCLC, approximately 1–2%, the proportion of NSCLC patients who receive molecular testing, increasingly high at academic centers, variable at community sites, the proportion who are in the second-line setting, have received prior platinum-based chemotherapy, the proportion who have not received prior RET inhibitor therapy, and the proportion of eligible patients who consent. The resulting eligible population at any individual site may be 2 to 5 patients per year, requiring a large, well-distributed site network to achieve the target sample size within the planned timeline. Screen failure analysis, identifying and addressing enrollment barriers. A screen failure occurs when a patient enters the screening period, signs the ICF and undergoes screening assessments, but does not proceed to randomization because they fail to meet one or more eligibility criteria or because they withdraw consent during screening. Screen failure analysis, tracking the reasons for screen failure by site and by eligibility criterion, is essential for identifying modifiable enrollment barriers. In Clarity 301, a 31% screen failure rate is clinically significant. 3 out of every 10 screen patients are lost to screen failure, reducing the effective yield from the site network. The most impactful screen failure categories: insufficient NGS sample, ambiguous RET fusion result, performance status decline. Each point to different interventions. For NGS sample issues, improving tissue collection protocols and adding liquid biopsy options. For performance status decline, accelerating the time from referral to consent. For prior RET inhibitor use, updating site training on the evolving treatment landscape. Trial timeline management, the enrollment curve and milestone tracking. A Phase 3 trial's enrollment timeline is a critical path that determines when the last patient is enrolled, when the target number of primary endpoint events is reached, and when the NDA can be filed. The enrollment curve, plotting cumulative patients enrolled against time, is monitored against the enrollment plan established at trial inception. Deviations from the planned enrollment curve trigger escalating interventions. Underperforming sites receive increased CRA monitoring visits and coordinator support. New sites are added if the network is systematically underperforming. Geographic expansion, adding sites in ex-US countries with higher eligible patient density, is pursued if US enrollment is insufficient. For Clarity 301, the decision to expand to Japan in year 2 of enrollment was driven by a systematic analysis of U.S. enrollment performance, screen failure rates, and the competitive trial landscape. Japan's high NGS testing rates and efficient academic oncology networks made it an operationally attractive expansion target. Patient retention and protocol compliance. Recruitment is only half the enrollment problem. The other half is retaining enrolled patients on study. In an oncology trial with a chronic oral investigational drug, velaratinib is taken once daily. Patients must comply with dosing schedules, attend study visits at 8-week intervals for imaging assessments, complete patient-reported outcome questionnaires, and report adverse events promptly. Patient withdrawal, for reasons including disease progression, which ends efficacy follow-up, drug-related toxicity, which may lead to protocol-specified dose modification or discontinuation, death or patient decision, affects the primary endpoint event count and the statistical power of the trial. Patient retention strategies include clear communication of the study visit schedule at consent, minimizing the burden of study visits, co-locating imaging and clinic visits, allowing telemedicine for certain assessments, proactive safety management, addressing toxicities early to prevent drug discontinuation, and coordinator engagement, regular check-in calls between scheduled visits for patients on investigational drug, competitive trial dynamics, and patient pool management. In a small molecularly defined patient population, the competitive trial landscape directly affects enrollment. If a competing Phase 3 trial opens at a site that is already enrolled in Clarity 301, the two trials compete for the same narrow pool of RET fusion-positive second-line NSCLC patients. The CTM monitors the competitive trial landscape using clinical trials, GOV, and direct feedback from site coordinators. Which trials are open at which sites, how many patients each site has enrolled in competing studies, and whether there are eligibility criteria differences that create non-overlapping eligible populations. Where possible, the CTM negotiates with competing sponsors to avoid direct overlap at high-volume sites. A practice called site scheduling or enrollment coordination that is common in small population oncology development programs. This does not involve anti-competitive behavior with respect to pricing or commercial terms. It is purely an operational coordination to ensure that site resources are not overwhelmed by simultaneous competing demands for the same patient population. Arventa hired a CRO, Pinnacle Clinical Research, to monitor Clarity 301. That does not mean Arventa handed over the trial. Osei, Arventa Global, Clinical Trial Manager. This is the most misunderstood dynamic in clinical operations. Sponsors outsource execution to CROs because CROs have the site relationships, the monitoring staff, and the global infrastructure that a mid-size company like Arventa doesn't have in-house. But the sponsor cannot outsource regulatory responsibility. If Pinnacle makes an error, a monitoring visit that misses a consent violation, a safety event that doesn't get reported on time, that's Arventa's problem with FDA, not Pinnacle's. So my job is oversight of the CRO, not reliance on the CRO. What that looks like: weekly operational calls with the Pinnacle project manager, reviewing every monitoring visit report within 5 business days, not accepting them, reading them. Tracking metrics: visit completion rate by site, outstanding query counts, deviation rates by site. Co-monitoring visits: I join the CRA at specific sites to independently assess the quality of monitoring. An audit of Pinnacle's monitoring records, annually. The delegation of authority log is explicit about what Pinnacle can do and what stays with Arventa. Medical monitoring, SAE assessment, regulatory submissions, Arventa. Site visits, data monitoring, query management, Pinnacle. The boundary is documented. A good CRO makes this trial possible, but the CRO is a contractor, not a co-sponsor. Agentforce CRO oversight tracking monitors monitoring visit timelines, metric dashboards, and escalation flags across the Pinnacle relationship. The CRO industry, structure and function. Contract research organizations, CRO, are companies that provide a range of clinical research services to pharmaceutical, biotechnology, and medical device sponsors on a contracted fee-for-service basis. CRO services span the full drug development continuum: early phase clinical research, phase 1 pharmacokinetic and safety studies, phase 2 Enrollment 3 trial management, regulatory submissions, data management and biostatistics, pharmacovigilance and laboratory services. Large global CROs, Covance, IQVIA, Parexel, PRA Health Sciences, Syneos Health, have site networks, monitoring staff, and regulatory expertise in dozens of countries, providing sponsors with the global operational infrastructure that would be cost-prohibitive to build in-house for a single development program. Smaller sponsors, like Arventa at the Clarity 301 stage, rely heavily on CRO capabilities. Large pharmaceutical companies may use CROs for specific functions, regional monitoring in countries where they lack staff, while managing other functions internally. The CRO market is highly competitive, and CRO selection, based on therapeutic expertise, geographic reach, site relationships, and quality track record, is a strategic decision for the sponsor. Sponsor oversight, the non-delegible regulatory obligation. FDA Regulations, 21 CFR 312. 52. Permit a sponsor to transfer any or all of its IND obligations to a CRO, but the transfer does not relieve the sponsor of responsibility for the conduct and integrity of the study. If a CRO's monitor fails to detect and report a consent violation, the sponsor is accountable to FDA. If a CRO project manager fails to ensure timely SAE reporting, the sponsor faces regulatory consequences. This regulatory structure creates the sponsor oversight imperative. The CTM cannot simply rely on the CRO to do its job correctly. The CTM must actively verify that the CRO is performing its contracted functions to the required quality standard. Sponsor oversight of a CRO includes reviewing monitoring visit reports, co-monitoring visits to assess CRA quality, metric tracking, visit completion rates, outstanding data queries, deviation rates, governance meetings, and periodic quality audits. A CTM who treats CRO management as passive contract administration, receiving deliverables without critically reviewing them, is operating below the required oversight standard. The delegation of authority, defining the sponsor CRO boundary. The delegation of authority, DUA, document specifies which of the sponsor's IND obligations have been transferred to the CRO, and to which specific CRO personnel. Clear DUA boundaries prevent gaps in accountability. If a task is not clearly assigned to either the sponsor or the CRO, it may not be performed by either. In Clarity 301, the DUA assigns to Pinnacle Clinical Research site monitoring visits and monitoring visit report preparation, data query management, site regulatory document collection and filing in the ETMF, and site training support. The DUA retains at Arventa medical monitoring, assessment of adverse events and their relationship to the study drug, regulatory submissions, IND amendments, protocol amendments, SAE reports to FDA, DSMB meeting support, and protocol interpretation. The authoritative answer to any site's protocol questions comes from Arventa, not Pinnacle. CRO staff listed in the DUA must be named individuals, not just role descriptions, and the DUA must be updated when CRO personnel change. Monitoring visit reports, the CRO's operational deliverable. The monitoring visit report, MVR, is the document prepared by the CRA after each on-site monitoring visit, summarizing what was reviewed, what was found, and what actions are required. An MVR covers the documents reviewed and their status, the data reviewed and any data discrepancies identified, protocol deviations identified and their classification, minor or major, SAEs reviewed and their documentation status, consent forms reviewed, and any site issues that require sponsor or site action. The CTM's review of MVRs is not a formality, it is active quality oversight. A CTM who reviews an MVR and identifies that the CRA failed to review consent forms for the last 3 enrolled patients, or that a major deviation was classified as minor, must escalate to the CRO project manager and ensure the issue is resolved. Systematic patterns in MVR findings. A site with persistent consent documentation issues, a CRA whose reports consistently miss SAE review, indicate either site quality problems or CRA quality problems that require different interventions. Risk-based monitoring, the modern approach to site oversight. Traditional clinical trial monitoring relied on 100% source data verification, SDV, a CRA visiting the site and comparing every data point in the EDC against every source document, patient chart, lab report, imaging report, to verify accuracy. Risk-based monitoring, RBM, introduced by the FDA and EMA in guidance documents in 2013, replaces 100% SDV with a risk-stratified approach, identifying the data elements and sites that carry the highest risk for patient safety or data integrity, focusing monitoring resources on those risks, and using central statistical monitoring of data trends to identify sites with anomalous patterns that might indicate data quality problems. For Clarity 301, the RBM strategy focuses SDV on primary endpoint assessments, PFS determining imaging reads and their timing, safety data, adverse event documentation and SAE reporting, and eligibility determining assessments, NGS reports, performance status assessments at all sites, with additional focused monitoring at sites that show anomalous patterns in central monitoring, unusually high or low event rates, unusual query response times, unusual data entry patterns. The monitoring visit report from the MD Anderson team came in Friday. 2 deviations flagged. I spent the weekend reading the source documents. Osei, Arventa Global, Clinical Trial Manager. A protocol deviation is any departure from the approved protocol. The question is always, what kind, and what does it mean for the patient and the data? Deviation 1. A CT scan performed at day 64 instead of the protocol-specified day 56, plus or minus 3 days. The window is 53 to 59 days. Day 64 is outside the window. The patient's imaging date will be excluded from the primary PFS analysis per the statistical analysis plan. The patient remains on study. The investigator is counseled. This is a minor deviation, administratively significant, but no patient safety risk. Deviation 2. A patient was enrolled with an NGS report that listed RET rearrangement without specifying the fusion partner. The inclusion criteria require RET fusion confirmed by a validated assay. Our central lab is reviewing the archived tissue now. If the central lab cannot confirm, this patient may be excluded from the primary analysis population. That's a major deviation. It affects eligibility and potentially patient safety if this patient doesn't actually have a RET fusion. Every deviation is documented in the EDC, assessed for impact on patient safety and data integrity, classified and logged. Major deviations are reported to the IRB and in the IND annual report. Systemic patterns, the same deviation at multiple sites, trigger a root cause analysis and corrective action. Agentforce deviation management tracks deviation rates by site, classification, and COPPA status across the trial. Protocol deviations, definition, and classification. A protocol deviation is any departure from the approved protocol, IRB-approved procedures, or GCP requirements that occurs during a clinical trial. Deviations are classified by their potential impact on patient safety and data integrity. Minor deviations have no meaningful impact on patient safety or the reliability of the data. A visit that occurs 1 day outside the protocol window, an ECG performed at the wrong time of day, a lab sample collected in the wrong tube type that was repeated correctly within the assessment window. Major deviations have potential or actual impact on patient safety or data integrity. An enrolled patient who did not meet all eligibility criteria, a safety assessment that was not performed when required, an investigational drug dispensed to the wrong patient, a patient who received study treatment without signing the informed consent form. Major deviations are also called important protocol deviations in FDA terminology and must be reported to the IRB and included in regulatory submissions. The CTM is responsible for ensuring that all deviations are identified, assessed, classified, documented, and managed according to the protocol's deviation management plan. The impact of deviations on primary endpoint data. In a randomized controlled trial, protocol deviations can affect the statistical analysis in multiple ways depending on the pre-specified analysis strategy. In Clarity 301, the primary analysis population is the per-protocol population. Patients who were eligible, received the study treatment, and had primary endpoint assessments within the protocol-specified windows. Patients whose CT scans fall outside the assessment window are excluded from the per-protocol analysis and contribute only to the intent-to-treat analysis. In most regulatory submissions, the primary efficacy analysis is pre-specified as either ITT all randomized patients, regardless of protocol compliance, or per protocol, patients who received treatment and had valid assessments, with the other analysis serving as supportive. The choice of primary analysis population, agreed with FDA in the SBA and pre-specified in the statistical analysis plan, determines which deviations affect the primary efficacy claim and which are operationally significant but statistically irrelevant. The CTM must understand this distinction to prioritize deviation management resources appropriately. Corrective and preventive action, the quality system response to deviations. When a deviation is identified and documented, the sponsor and site must implement a corrective and preventive action, COPPA, plan, a structured response that addresses both the immediate correction, what is being done to fix the specific instance, and the prevention, what systemic change is being made to prevent recurrence. A COPPA for a consent timing deviation might include immediate correction, ensuring all current screening patients have the consent discussion before any screening assessments are performed. Root cause analysis. Why did the site consent after screening? Inadequate training, workflow confusion, scheduling pressure, and prevention. Updated site training on the consent process. A checklist that coordinators must complete before initiating screening assessments. COPPA plans are submitted to the IRB when the deviation triggers IRB reporting requirements, maintained in the TMF, and reviewed by the sponsor and CRO to verify that the prevention measures have been implemented and are effective. Repeated deviations of the same type at a site after a COPPA has been implemented indicate that the COPPA was ineffective and may signal a systemic site quality problem requiring more intensive intervention or site closure. Reporting deviations to regulators, IRB, FDA, and the IND annual report. IRB reporting requirements for deviations vary by institution and IRB policy. Most require immediate reporting of major deviations involving patient safety risks, a patient who received the wrong drug, a patient enrolled without consent, and aggregate reporting of minor deviations in the annual continuing review submission. FDA does not require immediate reporting of individual protocol deviations, with the exception of deviations that meet the definition of a serious adverse event or an unexpected serious adverse drug reaction. However, the IND annual report, due to FDA 1 year after the IND effective date and annually thereafter, must include a summary of deviations and a discussion of any patterns or trends identified. FDA inspectors reviewing an IND annual report that shows a high rate of deviations at specific sites, or a pattern of the same deviation type across multiple sites, will treat this as a signal of systemic quality problems in the trial and may prioritize those sites for FDA inspection. Site closure for quality failures, the CTM's escalation authority. When a site's deviation rate exceeds acceptable thresholds, when a major deviation indicating systemic quality failure is identified, or when a site is found to have fabricated or falsified data, the CTM, in consultation with the clinical program team, regulatory affairs, and legal, may initiate site closure. Site closure involves notifying the site PI and IRB, suspending enrollment of new patients, completing safety follow-up for enrolled patients, who may need to be transferred to other sites or treated off-study, retrieving investigational drug, and archiving regulatory documents. Sites closed for quality failures are reported to FDA in the IND annual report and may be flagged as for cause closures, a designation that can affect the site's reputation in the clinical trial community and its eligibility to participate in future studies. The decision to close a site is not taken lightly. It reduces enrollment capacity and may damage relationships with key investigators. But it is the sponsor's regulatory obligation to ensure that data submitted to FDA in support of an NDA is reliable and generated in compliance with GCP.